Max Jaderberg, who leads AI at Isomorphic Labs, the company that came out of DeepMind, known for making the series of AlphaFold products. I've been trying to get Max for quite a while, so very excited to hear the latest and greatest. And with that, I hope it inspires many more of you to work into biology. I think it's probably one of the most exciting domains out there. I'm very biased with that, but welcome, Max.
All right, thank you. It's really great to be here. Thank you, Nathan and team, for organizing such a wonderful event. I'm going to be speaking today about what we're working on at Isomorphic Labs. Isomorphic Labs, as Nathan mentioned, originally a spin-out out of DeepMind, headquartered here in London. We've been going for about 3.5 years, coming up to 4 years, and really laser-focused on this problem of how we can revolutionize this world of drug design. But actually our mission is a bit bigger than this, and it's really just a simple mission that we think about, and that's just simply solve all disease. But we actually, it's a pretty audacious mission, but we mean this very seriously. And we know this is not going to happen overnight, and this is going to take a long time. It's also not going to happen in isolation. It's something we're not going to be able to do alone. And we're going to have to really mobilize a whole industry, actually many industries, to get behind this and march towards this. But I think the really exciting thing from my point of view is that we can really see that this new technology that we're all developing now, machine learning, AI, these incredible models, are actually pointing us really strongly in this direction. So to start off, I want to go back to some of the origins of the company. And actually, it's the same origins as myself. I've been working at the forefront of deep learning research for about 15 years now, and I spent a long time at DeepMind. And DeepMind is one of these amazing places where, when I was there, we were really working on defining and pioneering this methodology called reinforcement learning. And reinforcement learning is incredibly useful now and back in vogue with the amazing ability to align these large language models, create these amazing reasoning style behaviors emerging in these large language models. But what is reinforcement learning? Well, at the core, it's this ability to learn from actions in some sort of environment, some world. That could be the real world, but most usually where we find this cropping up really well is in virtual worlds. And at DeepMind, we spent a long time working on these methods, working on games like chess, and then more and more complicated games like StarCraft. These are very rich, multiplayer, very complex dynamics. And the crux of reinforcement learning is that, unlike the traditional supervised learning approach, in supervised learning, for every question you ask your neural network during training, you have to have a concrete answer of what the right answer is. And that's how you train your network. And reinforcement learning gives you the ability to train these networks without knowing what the actual answer is. But all you need to know is whether the answer the neural network spits out, whether it's good or bad, and ideally how good or how bad this is. And this type of framework unlocks so many new problems. So we were applying this to games. And I think, for me, one of the most exciting moments that showed the efficacy of reinforcement learning was this ability to master the game of Go with a program called AlphaGo. Go, for those who don't know, is a very, very complex board game. It's one of the oldest board games played. And unlike chess, where chess is a game that there are actually not that many states of a chessboard, and few enough that you could beat this in the late 90s with something like Deep Blue by just brute forcing solutions here, tree search style solutions. But Go, there are just too many possible states, and so you have to resort to deep learning, to reinforcement learning, to actually learn these solutions and these agents. And the AlphaGo story culminated in this amazing competition match with one of the best players in the world, Lisa Doll. AlphaGo went on to win 4-1, which was great, but actually, for me, the most exciting thing that came out of this AlphaGo story was what I call Move 37, in game 2 because it was literally the 37th move in game 2 of this series against Lee Sedol. And you can see the move up here. I'm not a Go expert, but as you saw this unfold, people were shocked about this move that AlphaGo played because it actually looked like a mistake. People thought it was a mistake. It was a move that had never been seen in the history of Go play before. And it was very, very strange. And as I said, it looked like a mistake until, as you unrolled the game many moves later, this became the critical move that secured the win for AlphaGo in that second match. And so actually, what looked like a mistake from artificial intelligence was actually a moment of real creativity and invention. And so the question is, why am I talking about games when at ISO we're thinking about solving all disease? And well, what we want to do is move from this world of games where we've actually developed a lot of these core deep learning and machine learning algorithms into the world of real science and really see these moments of invention and creativity applied to the world of science. And well, why this is hard, why science is hard is because science exists in actually the real world. It's not a virtual world. And the real world is really, really messy. And the real world is hard to interact with because there's just one of the real world. Time unfolds painfully slow. You can't run many, many things in parallel unless you have physically many, many things. And so what we need to think about doing a lot is creating these AI analogs or world models of the real messy world. And then this gives us a substrate, a virtual substrate that we can experiment in and start to do science purely on a computer. Now, we've been able to do this a lot in the past in the world of physics, because in the world of physics, we can actually write down equations in maths that can really nicely and accurately describe things like the unfolding of the universe many millions of light years away, even though we're just here on Earth. And somehow these equations that we've managed to find generalize very, very far in this world of physics. But when I think about solving all disease and drug design, we operate in the world of biology. And biology is just phenomenally complex. If you think about the dynamics of the cells, there's just so many crazy nonlinear dynamics that it's basically impossible to write down an equation by hand for how the cell works or the dynamics of the cell. You can perhaps write down the equation of how two individual atoms of a cell might interact, because that's just physics. But then actually scaling up, the simulation of those atoms to the trillions of atoms in the cell is just completely unfeasible. And this, of course, is where the world of AI and machine learning comes in. And we believe at Isomorphic that there exists this isomorphism that we could create between the biological world and the world of machine learning and AI and information science. And that's what we're laser focused on really realizing the potential of. And why this is so important for drug design is that actually the number of new drugs per $1 billion of R&D spend that are coming out is actually exponentially decreasing. So this is a log axis on the y-axis, which means it's becoming harder and harder, more and more expensive to create new drugs. But then on the other side of the coin, you've got things like Moore's law, where actually the amount of compute power that we have access to is actually just exponentially increasing. And probably these days it's not Moore's law we should care about, it's Jensen's law. But if you look at GPU flops, these are also just exponentially increasing year on year. And so really the opportunity that I feel day to day is how do we bridge these two worlds? And can we reverse this curse of Eroom's law and jump on this exponential wave of compute power that we have access to? And to do that, we're going to be creating AI analogs of this biological world, bring more and more of this onto a computer. And I want to dig into a particular example of how we're doing that at Isomorphic Labs and zoom into the body and think about the world of proteins. And proteins they're the fundamental building blocks of life. And they create all of these molecular machines inside of us. And for drug design, what we want to do is actually try and modulate the function of these proteins. And if we can modulate the function of a protein, we're well on our way to designing a new drug. So what are proteins? Well, proteins are a sequence of amino acids. Each one here is depicted by a different letter. There's about 20 different amino acids inside of us. What are amino acids? Well, amino acids are molecules. They're collections of atoms, and these amino acids, these molecules, they join up into a long sequence, a long string, and that's a protein. But these proteins don't exist as strings. They actually fold up inside of our cells and create these amazing, beautiful 3D shapes, 3D structures. And it's really these 3D structures that are that give these proteins their particular functions and allow them to operate together with each other as molecular machines. Now, the problem is that understanding the structure of these proteins and therefore how these proteins work is really, really hard. And so traditionally, the only way to do that was to actually try and work out these structures experimentally, actually observe them experimentally. So what does that mean? This is real experimental lab work that takes months, maybe even years. For some proteins, people haven't figured out how to experimentally derive the 3D structure. It's painstaking work. You have to actually synthesize your protein, purify it, then you have to crystallize it, literally grow crystals, then shoot X-rays at this and observe the X-ray scattering, and then somehow infer the 3D structure from that. It takes a very, very long time, but people have been doing this over the last 50 years and built up a database of a couple of 100,000 protein structures. And then, incredibly, in 2020, 2021 was the breakthrough from DeepMind of AlphaFold 2, which took all of these existing crystal structures and trained a deep neural network to take as input the input sequence of amino acids, and as a result, this network could very, very accurately predict the 3D structure of proteins. This was a huge breakthrough, and for the first time, this allows us to understand the structure of proteins without doing any experimental work. And so what you can do is start to fill in the gaps of the known protein universe. And so what we did was take AlphaFold 2 and run it across all 200 million proteins that we knew about at that point in time, and predicted the structure of these and actually released these into the world alongside the model. And incredibly, it's estimated that the amount of experimental work that was saved with predicting those 200 million structures is equivalent to about 1 billion years of research time, which is just crazy. Just creating one of these structures experimentally can take a whole PhD's worth of work. And of course, this work was recognized with the Nobel Prize in Chemistry just last year, and it's been incredible to see the journey of that. So now, back to drug design. So we have these proteins, but these proteins, they don't exist in isolation. They're molecular machines, but they don't work alone. These proteins work as machines by interacting with other biomolecules, other proteins, or other biomolecules like DNA, RNA, small molecules, things like that. And so in this example, you've got a DNA repair protein, and it works by interacting with DNA. DNA comes in, the protein clamps shut, and that facilitates DNA repair in this case. And so then the question is, what is this drug? Well, a drug is another type of molecule. That could be another protein, or sometimes small molecules. And a drug is something that will come through the body, in this case a small molecule, and it will stick to a protein, for example this one just over here. And through doing that, it subtly changes the shape of this protein, or blocks this protein from doing something that it would normally do, and it changes the function. In this case, it actually stops DNA repair. In this particular example, that's quite important in cancerous cells, to stop these cancer cells proliferating. And so then the question for us at Isomorphic Labs is, okay, if we want to design a drug molecule like this that modulates these molecular machines, how can we do that really rationally? How can we understand these systems so we can do this really rationally? And so to do that, we would need an AI analog, a model that understands how all of these biomolecules come together. And last year, we had a breakthrough with this at Isomorphic Labs. We developed a new model called AlphaFold 3, which for the first time allows us to understand very accurately the structure of these proteins as they come together with other biomolecules like DNA, RNA, small molecules. How does this model work? Well, this model takes as input the sequence of amino acids, the protein sequence, but then it also takes as input the DNA sequence, the bases there. It also takes in as input the atoms of the molecule that could be a drug here. This is a neural network with a large processing trunk. This trunk is based on transformers, but unlike, I'm sure you know transformers from large language models, but unlike the use of transformers with large language models, where these transformers there operate on one-dimensional sequences of tokens, that is your sentences and language, in our case, we use what we call a pair former, which operates on this two-dimensional interaction grid, which allows us to model explicitly every pair of possible interactions that could occur between the different molecules in this biomolecular system. So we have this large processing trunk, and then the features from this pair former, they condition a diffusion model. And again, we know these diffusion models from the amazing image generative models that are out there. In these image generative models, people are generating the pixels of an image. And just like generating the pixels of an image, instead our diffusion model is generating the 3D coordinates of every single atom in this biomolecular system. And this is really, really powerful because now we have a model, an AI analog of this biomolecular world where we can actually change the design of our drug, and we can see how changes in the design of that drug instantly will affect the 3D structure of this protein, affect how this drug interacts with this protein, and it now allows our chemists to start rationally making these changes. And the nice thing is our chemists can actually start to trust these models because these models are getting very accurate. Here are some results on a benchmark which shows the ability, the accuracy of predicting small molecules, ligands, with proteins. So small molecules are a really important class of drugs. These are drugs that normally you can take orally as a pill. And you can see that AlphaFold 3, which came out last year, is far more accurate than the previous methods in this space. And actually, these results are a year old. At Isomorphic, we never stop working on this core problem of structure prediction, and there's always advancements to be made here. But excitingly, with this new class of model, we find that actually we improve our understanding of even how proteins interact with each other, including some really, really important areas like on the far right, protein antibody structure prediction. So antibodies are another really important class of protein-based molecule, which is another class of therapeutics that's really important. And you can see that AlphaFold 3 doubles the accuracy of predicting these structures with respect to the previous state of the art, which was actually AlphaFold 2. So I want to show you how this model can be used in practice. And here's an example. We've taken actually a piece of published drug design work and recreated all the steps with AlphaFold 3. This is trying to drug this orange pocket on this TIM3 protein. And here, the authors have actually designed this blue molecule that fits just here. And you can see the AlphaFold 3 prediction in color with respect to the ground truth in white. And AlphaFold 3 very accurately predicts the structure. Then the designers of this drug, they keep adding atoms to this molecule to make it bind and stick stronger and stronger. And at each step of this design process, we can query this structure prediction model and see how subtle changes in the design, like adding a new ring, can subtly change the orientation and help us understand better and better what could be a stronger and stronger binding drug. And the cool thing is that this is now the reality that our chemists in London are living out day to day. So we have a whole suite of models, structure prediction models like AlphaFold 3 are really at the core, but we have many other models that predict other properties of these biomolecular systems, things that are really important for drug design, and then have created a whole suite of software that allows world expert drug designers, medicinal chemists, computational chemists, biologists to then use the power of these models to design new drugs. And so we have this beautiful game that's being played between our human drug designers and now this virtual biomolecular world. It's a bit like a video game. And so the question is, if we can have human players playing this game against our virtual biomolecular world, well, why can't we start to train agents instead that play this game in our biomolecular world? Now, the really cool thing about this setup is suddenly all of the drug design is happening on a computer. And so the question is, okay, what happens if we have many, many computers? Well, we can just scale up the amount of design we do. And this is a real reality. This now allows us to imagine many, many possible different hypotheses, all in parallel, all being designed by agents in parallel. It can allow us to go after many different targets or possible mechanisms of action, all in parallel. So this opens up a huge amount. And we really see this play out in practice. This is not science fiction. We're doing this today. We have some very powerful generative models and design agents that are able to, as you can see on the left, take as input your target protein, what you want to modulate, and can produce these pink molecules, these small molecule designs that, in this case, are responsive to different mutations in that target protein. Of course, mutations in DNA and then proteins are often the root cause of some really serious diseases. And what you can see on the right are real experimental results. So we create these designs from our design agents, and then we take them into the real wet lab, and we see how well they work. And here, the higher the better. This is how active, how many of these molecules that are designed are active against what we want to try and drug. And the baseline here is what is traditionally done in the industry, which is what's called screening, where you take maybe hundreds of thousands, maybe millions, maybe even billions of molecules, and you experimentally test every single one of these molecules against your protein in the real lab. And you're randomly screening and trying to find some hits, and you probably get, if you're lucky, 1% to 3% of these will be hits. But you can see here, these generative models and design agents are able to get 20, 40, 60% hit rates, sometimes for easy proteins like on the far right. Even 100% of the designs come out as positive hits. So this is completely changing the way that our chemists are actually approaching drug design day to day. And these projects, these are not fictional projects, these are live projects that we run internally, also with our collaborators like Novartis and Lilly. The other really, really cool thing for me as a deep learner is that as we're modeling this space, as we're thinking about doing small molecule design, the underlying models like AlphaFold 3, like our latest structure prediction models, they understand the world in terms of atoms, not in terms of whether this is a protein or a small molecule or DNA. And because atoms are the fundamental building block of these models, we can actually apply them not just to small molecule drugs, but also to large molecule drugs, things like antibodies and nanobodies. And again, we've been doing this and starting to see really, really cool results here. So this is this same generative design agent, but now applied to nanobody design. And you can see on the left, this is creating a whole diverse set of designs for different target proteins in green. The designs are in pink. And again, we take those designs, we take them into the real lab, and we test them out. Normally in this space, again, the traditional methods would be screen millions of different designs and try and get maybe 1% of those coming out positive as hits. Here, we only need to take a couple of hundred of these designs to get really meaningful hits and really meaningful hit rates. These are exquisite hit rates with respect to what the industry is currently capable of doing. So it's a really, really exciting time for us, for our chemists, and I think also just the future of drug design, because we now have an ability, through all of these models that we're able to create, of really understanding these biomolecular systems better, and therefore rationally designing molecules against them to modulate them to go after some of the hardest diseases around. And so at Isomorphic, we really think at the core about creating these world-leading structure prediction models of biomolecular complexes, of biomolecular systems. And we couple that with these really, really powerful generative models, search methods, reinforcement learning, to deliver ultimately really high hit rates, ways of modulating these proteins. And then again, the really, really exciting thing is that these models are general and they're modality agnostic. They're general in the sense that we can go after any target protein, any disease area, any indication, and we can be producing small molecule drugs, but also large molecule drugs, things like antibodies. And so what we're seeing out here in one particular corner of the scientific universe is this ability of taking the real messy world and creating these AI analogs, these models of the real messy world, and then we can train agents to use this as a game environment and play the game of science all on a computer and do in silico on a computer open-ended scientific discovery. But this is not just for fun. We create, real results, real artifacts that we go out and synthesize and create in the real world and effectively change the real world, hopefully for the better. And so I think this paradigm will play out, not just in the drug design space, but actually all across of science. So it's a really, really exciting time for the field. And I just want to finish this talk with one last thought, which is going back to this AlphaGo moment, and maybe a question for us to think about is, well, okay, we had this amazing move 37 moment in the game of Go, and it's all well and good, but ultimately Go is just a game, right? But then what does the move 37 moment look like when we're doing the game of drug discovery? Or actually, what does the move 37 moment look like for any other part of science? And I think these are really interesting questions to think about and to start observing as we see them happening. And the really exciting thing from my point of view is I'm catching glimpses of that happening internally day to day, where we have designs of molecules that are created by these agents that actually some very experienced medicinal chemists with decades of experience behind them look at them and say, no, this will never work. And then we go into the lab and we make them, because obviously we have to trust our models, and we find that they actually work. And they work maybe in a way that we never expected, that's never been published in the literature before. And that surprises everyone, both the chemists and us machine learners. So we're catching glimpses of this, but I think there's so many more of these to come. So that's all for me. Thank you very much.